record button, and it even tells me it's recording. Awesome. So we've got a couple more minutes. We'll see if anybody pops in. But even if it's just you two, that's more than I've had in the past. So I did open up this to my seated class, even though I have one of my seated classes in lab right now. So I know none of them are going to come, but they were worried about reviewing. I think you're all fine. I have strong faith in everyone's ability to complete an exam um, without any issues. So I'm going to go ahead. I know it's not quite 11:30, but I'm going to go ahead and get started here. We only have like 40 minutes. So this is your first exam for Bio 110, and it will be taken on Canvas. I do use matching, fill in the blank, and short answer. Grading, like the system will automatically grade matching and fill in the blank questions. But if you misspell something, I always go through and check that. And I also check the matching to see if like everyone is missing a particular question, then I would make some adjustments to the point values on that assignment or that particular question. And I like doing ordering questions too, especially when we're talking about things like the biological organization from smallest to largest or the steps of the scientific method rather than having you write it out. A lot of times I have it in there as ordering. And then short answer questions. The biggest thing that I see when people are answering short answers is that they panic. Okay. I don't want that. Like most of my short answer questions are going to be the like check your knowledge questions, that sort of question that you see. Oh yeah, Caitlin's here too. Katie. All right. So most of the things that you'll see in short answer are going to be like the check your knowledge questions, like the stuff that you see in your homework assignments. For my online students, like what you see on those attendance quizzes when I'm asking you some of those answers or some of those answers. And what I've been getting back is fantastic. Okay. Um, so, one of the things that I do want to make note of now, because it will become more and more important as we move into the semester, is those short answer questions. You want to be specific in your answer. So, if I ask, and I like to ask questions with like parts to them. So, if I ask, for instance, are viruses alive? Why or why not? You can't just say yes or no because I'm asking you to support your reasoning. And that's true for any science class. That's what we're practicing in biology for the most part is thinking critically and supporting your reasoning. And by supporting your reasoning, that's what's telling that you understand the topic at hand. So when you see short answer questions, a lot of times people run into the problem of like not answering it completely. So make sure that you are reading the question completely. Make sure that you are looking, if it says like explain, support, why, how, what, I'm looking for more specifics. I want a little bit longer. And a lot of times those questions are worth more of a point value. If it says list, please don't write me a paragraph. Literally just write me like in a list, a bullet list or a numbered list. That is completely acceptable to me. I don't want to have to be digging through your paragraph to find that answer because I'm going to be grading uh 60 tests for this class for this section for this test. So that's the way that works. I like doing it on Canvas because I do get to hide names and grade by question. And so it allows me to be really critical of the question itself rather than like the student answers that I'm getting. Um, for my online students, you are in class as if it were a Monday, Wednesday class. The tests are meant to take no longer than 50 minutes. So there's usually a little bit more time on the online class because the Respondus browser does tend to slow things down a little bit. But you guys shouldn't need any more than 50 minutes to complete this test. Okay. Um, Look at all you guys popping in. I like this. This is the most students I think I've had in an online review session um, in years. So, yay. um, one of the things that I am going to say is that you will be asked to make a table in Canvas, and to make a table in Canvas is fairly easy. Um, there will be a toolbar. You'll click on Insert Table, and then whatever size table that you need. So that's just something that is going to be there. We're going to make tables a lot through the whole semester because tables are a great way to organize our thoughts and information. So I put down a quick thing. So my students who were in my seated class yesterday, we talked about what was the big topics. Like if I were looking at it and I was saying, okay, I need to know the information on these topics and then, be able to explain them in complete sentences. So for chapter one, you need to know the 6 characteristics of life and why those are important. For chapter one, you need to know the scientific method and the difference between a hypothesis and a theory. For chapter two, it's the chemical bonds. And I know everyone gets stressed about the chemistry, but I'm asking you to do matching on the definitions of the chemical bonds. So if you can identify that giving an electron and receiving an electron is ionic, then you're going to be good because when we do matching, we look for the keywords that tell us what we're seeing. You should always read the full answer options, but you should always also go, do I see keywords that without even looking at the list of things I'm matching to, do I just know what these definitions mean? And that's one of the things that I see a lot people getting stuck on. You don't have to, you know it or you don't, and you can figure it out if you don't. Like, right, get the ones you know down, and then you have like a 50-50 option for the ones you're missing, for instance. You need to understand the properties of water and like know that cohesion and adhesion and surface tension and such are properties of water, biological, what we're looking for. But the biggest thing is how the chemical properties of water, like the polar covalent bonds and the hydrogen bonds, result in cohesion and adhesion and surface tension. That is a question on the test, um, like an 8-point question on the test. So that's on there. For chapter three, there's the macromolecule table. You need to be able to tell me the elements, an example for each, the monomers and the polymers for each of the 4 macromolecules that we've talked about. You guys have been doing that throughout your studying, so you should be okay with that. Understand the difference of organic versus inorganic and why carbon is so versatile because carbon plays a role in all of these macromolecules. But there is a macromolecule table on the test. That is something that is on the test. And then with chapter four, what structures do all cells have and how is that related to evolution? Like, why do we see that as an evolutionary trait? And then you need to know the organelles and their functions. That is, again, going to be matching. I'm not going to give you a picture of a cell and be like, hey, label these. No, I'm not doing that. Okay. I have had way too many visually impaired students to do that, but matching is going to be a thing that you'll see on that. So if you can match the organelles to their functions, you should be good. All of this is not overly crazy. I think based on the assignment answers I've been getting in my online students and the answers I've been getting from my seated students, that you guys are all very well prepared to take this exam. Okay. With that being said, some of the major things that are usually popping up for people as far as things they want me to review for Unit 1 are the chemical bonds. Okay, people are comfortable with the characteristics of life usually. They're like, oh, yeah, we got that. That's what makes something alive. I'm cool with the scientific method. But it's the chemical bonds, the properties of water, that polarity and the hydrogen bonds, the biomolecule table, and then the cells and organelles. Is there anything else that you guys have that you're like, let me make sure Ms. Larson knows that I need to go through this? And you can always like unmute yourself and say it if that's easier than typing it.
Miss Larson,
Yes.
I had a question about the chemical bonds, just
Hmm.
because I was reviewing them, the nonpolar and polar covalent bonds in specific. It might be easier if I show you. Hang on, let me turn my video on. Can you see me?
Yes.
Okay, so I watched the lecture video where you went over chemical bonds, and I drew the example, and basically they're all electrons, right?
Hmm.
And what's confusing me is why it means no charge or charge, because when I think of electrons, I think they're all negative, therefore they're creating a negative charge. And when I think when they're when they combine together and they make a charge, it's like a proton, if that makes sense. I was wondering if you could kind of go more in depth to make it more understandable.
Yeah. So this is the slide I have for that, and I have this picture for this. So when you're talking about the charge, it's a matter of remember who's holding on to the electron tighter. Okay. I really do like the idea of tug of war because we've all seen somebody playing tug of war, right? And when you play tug of war, there's usually that rope in the middle that's marking like who's going to win. And the rope has to get to one side or the other for that person to win. When we're talking about covalent bonds, they're sharing electrons. And yes, electrons are negatively charged, okay? Protons are positively charged. But the proton acts as the pulling source, the individual who is on that rope pulling it to be, tug of war. So when we're looking at non-polar, okay, both oxygens, for instance, will share electrons. Okay, oxygens are going to share electrons to get to that octet rule, but neither oxygen's proton amount is larger than the other one. They're both equal because they're identical twins, right? Both oxygens are the same. And so while they're sharing that electron, they are playing tug of war, but neither one is pulling the electron closer to itself. So the electron is kind of just in the middle, and it doesn't end up resulting in any sort of positive or negative charge. And that's just, they're just hanging out in the middle. So neither way is that tug of war rope getting pulled. It's just staying in the middle. So no one's winning. There isn't any sort of like plus or minus on either side of that tug of war. It's just in the middle. But when we talk about something like a hydrogen compared to an oxygen, okay, one oxygen is going to have 8 protons, which are positive, and they have 8 electrons. The hydrogen has 1 proton and 1 electron. The hydrogen cannot pull on that electron as tight as the oxygen can. So when we think of that tug of war again, that's when you have the larger person on one side who is pulling on that rope and the rope is moving towards them. They are going to win that tug of war, but the other person doesn't give up. They don't let go. They dig their heels in. They're holding on. And so the electron isn't like completely lost. The larger molecule doesn't take the electron away. They're still both holding onto that rope, but it's just a matter of who's pulling that middle section closer to themselves. When I pull an electron closer to myself, right, I am pulling the electrical part um, towards myself, which makes me slightly negative, right? Because I'm gaining some of that electrical like negativity. And then since I'm pulling that closer, the other particle is going to be slightly positive because it's losing a little bit of that electrical charge. It's not completely gone. And it's not like it's the ionic bond where it's a complete positive and a complete negative. Instead, it's like we're still holding on, but like that rope, that electron is that middle of that rope of tug of war. It is closer to the larger person. And so they are winning, but in winning, they become slightly more negative because they're gaining more of that electron. Okay. Does that make sense? You have to you have to figure out a mental picture. For me, the tug of war works because I wish my chemistry teacher had taught me that. I think I would have done better in chemistry if I had that as my basis. Because when we're talking about it, like that makes sense, right? That's why water has that slight charge because oxygen is so much bigger. It is just hoarding those electrons closer to itself. And then the smaller molecule is holding on, but it doesn't get any of that electrical, that negative aspect. The oxygen gets all of that, which then results in the hydrogen being slightly positive. When we talk about ionic bonds, though, again, sodium doesn't want this single extra electron. Everybody wants to mimic the noble gases, right? Everything wants to have that octet rule fulfilled. And I think I made a reference to Labubus yesterday about it because I don't understand Labubus, but everybody wants one, right? It's just a fad. But in this case, in your elements, the fad is to have that octet rule fulfilled. And in order to do that, you may need to give up something that you have. But you're okay with that because now you're mimicking the things you're trying to be like. So when sodium donates the electron, it actually has lost one of its negative charges. And if I take away from a neutral thing, I take away a negative charge, I'm going to have a positive charge left over. Whereas if I have something that's a neutral charge and it picks up that negative charge, it's going to become more negative. So that's why ionic bonds, you have sodium, which is positive, and chloride, which is negative, because they've actually gotten rid of the electron. And there is a reaction there, like there is an energy transfer. You can see if you put sodium in pure water, like pure sodium in water will explode and light on fire. And that's because those electrons are leaving so fast in such high amounts that you're actually seeing the visible light from that chemical reaction. Now, when we're looking at a single molecule, we're not going to necessarily see that. Okay. But there are energy exchanges that are happening there. Okay. So those are our ionic and covalent bonds. Remember, non-polar just means no charge. If we add non in front of a word, it means without. And then polar is going to have that slight charge. And again, that's just because they're holding the electrons either tighter or not as tight. And then hydrogen bonds will form between things that are polar and things that have a charge. So your water molecules form hydrogen bonds, your DNA forms hydrogen bonds. There's a lot of things that do that. Okay. Questions on chemical bonds, because that kind of lets me go through this. Now, I'm not going to give you this as a matching list. So your matching would be ionic bond, and you're going to match it. It's going to say something like one element donates an electron while the other element receives an electron, resulting in an attraction between charged molecules. That's the more like in-depth definition. But again, if I'm looking for keywords, donates and receives are just gives and takes. And I'm looking at what's happening with that electron, right? The same thing if it shares the electron unequally. It doesn't matter how I write that. We should be able to identify that a polar covalent bond shares the electron unequally. That's what you're going to see from matching. So you're going to have these four. You're going to have to match them to their definition or to what they are. I'm not going to give you like examples. I'm not going to throw magnesium oxygen up there in order to make magnesium oxide. Like that's tricking you. I'm not going to do that. I need you to know these things so that when we start talking more about the macromolecules, it makes sense. And then also when we get into cellular respiration and photosynthesis, we keep remembering that these things play a role in that. That does then lead me to my properties of water, where when I see a water molecule drawn out, you'll always see polar covalent bonds or non-polar covalent bonds drawn as solid lines because the solid line is indicating that the electron is being shared. When you see a dotted line, that's your hydrogen bond because it's an attraction between the charges rather than a sharing of the electrons. So that's going to be that big difference there. Water forms these hydrogen bonds because we never have just a single water molecule. If I have a glass, if I have my Stanley full of water, there are billions of water molecules in that glass, okay? And those water molecules are liquid because they're consistently breaking and reforming those hydrogen bonds. When we heat up water and we get it to the point of steam and boiling, what we're doing is we are breaking the hydrogen bonds faster than they can reform. Okay. When we freeze water, what we're doing is we're stopping the hydrogen bonds from breaking and it becomes stuck in that lattice shape, which is why ice floats and it's less dense than water. So these hydrogen bonds play a role in the properties of water, those chemical states, which is, you know, the solid, liquid, vapor, okay, but also in things like cohesion and adhesion, or in the interactions with ionic and polar molecules, because when we have charges, charges can interact with things and do stuff, because that's what charges do. Okay, so if we look at the water molecules, right, we need to be able to identify that this is a polar covalent bond, and then the hydrogen bond will form between opposite water molecules. Okay, and then if we look at those, I have some pictures here, just I'll kind of arrange them around. So let's actually start with the idea of this hydrogen bond in ice. So remember I said the hydrogen bonds are constantly breaking and reforming in liquid, in water. They're just, it's like milling around. You're talking to somebody and then you go talk to somebody else and then you go talk to somebody else. And it's just kind of like you're floating around the room and that's what water molecules do. But when we freeze them, we actually make it so that these molecules, these bonds cannot break anymore. They're stuck. And the reason that they're stuck is they're so cold that they can't move. These things are moving all the time. And so that's why we actually see ice being less dense than water because the water molecules get stuck into a pattern, where in liquid water, they don't have a pattern. There's no pattern there. Okay. So that also then explains how water has a high heat capacity because water has to take a lot of energy to break those hydrogen bonds and keep disrupting them. When the hydrogen bonds are disrupted, they reform as quickly as they're broken until we get to a certain temperature when the molecules cannot keep reforming as fast as they're being broken. We're overcoming that level of breaking and reforming. We're basically going beyond the capacity of what that water can do. And so in your body, this is great because this is what's keeping you at a regular temperature. When you sweat, that's part of like the breaking the bonds at a higher things is what's helping you cool down. But it's also why your body doesn't just like instantly boil when you walk outside and it's 115 degrees, because water doesn't change temperature that quickly. And again, that all has to do with the hydrogen bonds, which can only exist because of the polar covalent molecules. In addition, the reasons we have things like salt water is when I put sodium chloride in water, these water molecules are going to be attracted to the opposite charges. And so this sodium is going to get surrounded by oxygens and get separated from this chlorine, which is now surrounded by the hydrogens. And so we're breaking the hydrogen bonds here a little bit because those are now attracted to something else. But that is why we see salt water and other things that have charges, anything that's ionic or polar, interacting with water because the charges in the water get attracted to the other molecules. And water molecules are tiny. There are way more water molecules than there are salt molecules. So that's going to be one of the things that we see there. That's the universal solvent. Okay. So we've talked about how water is less dense as ice because the hydrogen bonds are stable, how water has high heat capacity because we have to overcome those hydrogen bonds in order to change the temperature of the water, how water can act as the universal solvent. Oh man, I'm gonna have to start another meeting. Okay. And then when we talk about adhesion and cohesion, okay, we can see that those water molecules are sticking to themselves through those hydrogen bonds and then sticking to the outside part because those also have a charge. And so your straw, right? Things are going to have a straw there and there's going to be like a connection and a reaction at that. Okay. This is one of my favorite comics because help, help, I'm dissolving, but bears are insoluble. That's easy for you to say. You're not polar. Polar bears dissolve. Okay. When we look at our, thank you, Lila. I think it's hilarious. One of the chemistry professors had that outside their door for a while and it made me cackle, so I had to add it. Okay. When we look at the biomolecules, this is the table that I'm talking about. There are different ways to build it and look at it. It is in Blackboard. There is a table there. Everyone should, at the very minimum, I'm going to give you that it's a carb, lipid, protein, and nucleic acid. Everyone should, at the very minimum, know the elements are CHO, CHO, CHON, CHOMP, and be able to give examples for those. Okay. I'm not worried about you knowing necessarily if they're hydrophilic and polar covalent now. That is something that we are going to bring up again when we get into cellular respiration and photosynthesis in the next unit, as well as diffusion and osmosis. So, unit 2. I do want you to know the monomers and polymers. Okay, so monosaccharide meaning one sugar, polysaccharide meaning many sugars. Lipids don't have a true monomer and polymer. Na is fine as well, but no true is the better answer because they are built of things. Amino acids and proteins are fine for your proteins, and then nucleotides and nucleic acids for monomers and polymers and nucleic acids. I am asking that we memorize this because we are going to continue coming back to this through the entire semester. And it's like memorizing your multiplication tables. Once you have it memorized, you can move into more complex math. Once we have these biomolecules memorized, we can move into more complex biological processes. So this is the majority of your question for chapter 3. It's a 16-point question. That's the biggest part of chapter 3, and it goes through, this basically covers all of those objectives from that particular chapter. Okay. One of my students in the past made a table out of it, like a memory game. So if you're working on studying and that's something that you want to do, that's cool. Um, Dehydration, synthesis, and hydrolysis. Remember that our words mean things. So dehydration means to remove water. Synthesis means to make. So dehydration synthesis is to make by removing water. And hydrolysis, lysis means to break. Hydro means water. So hydrolysis means to break by adding water. This is how we are making monomers and polymers. Our cells are doing this all of the time. You don't have to think about it, which is awesome. If you had to think about every process that happened in your cells, you'd go crazy. That's also from chapter 3. And then from chapter 4, the biggest thing is just understanding like that prokaryotic cells are a little bit different. They don't have that nucleus, but they still have a nucleoid region. And eukaryotic cells have a nucleus. In addition, all of these cells share these structures, the cell membrane, cytoplasm, and then the genetic material. Okay. My online students, you guys gave me fantastic answers to your attendance quiz question, asking about why these things are retained, like how they play a role in evolution. Um, these are very original aspects of cells. When cells first started evolving, we saw cells had a cell membrane, they had cytoplasm, and they had genetic material. And those things are so important to the cells themselves that they've been retained across time. If you don't have a cell membrane, you do not have a barrier dividing the inside and the outside of the cell. So that was necessary early on. Cytoplasm holds all of the goop, the nutrients, the organelles, the genetic material, whatever it is, the cytoplasm is holding all of that. And then the genetic material, if you don't have DNA, you're not alive. If you don't have the ability to make proteins, your cells cannot function. And so these three structures are really good evidence of that characteristic of life of evolution because they are found in literally all cells. There aren't any cells that are lacking in a cell membrane, cytoplasm, or genetic material. All cells that have been looked at have those component parts. With a caveat, red blood cells lose it, but they have it to begin with. Genetically, they have them, but then they lose the nucleus, and so they become denucleated, but they have them to begin with. And then the biggest thing with organelles, matching flashcards are the best way to do this. You're going to be matching what these organelles do, okay? So structures and functions. And again, what I would personally do is I would cover up, if I were looking at a matching question, I would cover up the answer. So I would cover up the organelles themselves. I would look at the list of options the instructor gave me, and I would be like, which ones do I know? So if I start looking through and I go, oh, I know that the nucleus houses DNA, then I would answer that one and it takes nucleus out of the running. Okay. If I know that mitochondria generates ATP through cellular respiration, because it's the powerhouse of the cell, then I would answer that and take off the rest of it. Okay. That's chapters 1 through 4. That's basically what we have for this unit exam. The majority of questions are matching fill in the blank, and then there's a couple short answer. And as we move through the semester, the short answer becomes more common because we start digging. This is more of a recall unit where I'm asking you to memorize and remember some things, and then we're going to do more application of these materials in later units. It's why we need this as a foundation. Okay. Any questions that came up?
I was wondering for the hydrogen bond,
Yes.
um, just like to clarify, the hydrogen bond is like basically what makes that cohesion.
Yes.
Okay.
Yeah. Hydrogen bonds make everything happen in the water. The cohesion, the adhesion, cohesion is water sticking to each other, and that's happening because of those hydrogen bonds. Anything else?
Thank you.
Yeah, of course. You guys are ready for this. Like, I promise you guys are all fine. I know people are like, I don't know. Homework has been great. Questions have been great, both online and in class. Um, I am not concerned. If I was concerned, I would let you guys know. I have no problem telling people when I go, we are not ready for this. Hold on, let's back up. You guys are ready for this test. You will be fine. This one is going to be hard just because it's the first test and we get anxious and nervous. Just remember to breathe. You know this stuff. If you get test anxiety, your brain's lying to you. And I know it can be really hard to tell your brain it's lying to you, but it is. Make sure you just take a moment, take a breath, read what is being asked of you, um, and don't get in your own way. Do your best to answer things. I love giving partial credit. I love if you just are like, I'm not 100% sure, but here is my best take. 1,000%. I will give you some credit if I can. If you're super way off base and I'm like, you didn't even listen to my lectures, then it's a different sort of story, right? But you guys are going to be fine. Don't panic. Study. I, unless there's anything else, I think we're out of time too. So if you have any more questions, just send me an email, let me know. Okay. Bye, guys. Thank you.